Welcome to Biota Live. I'm Tom Barbalet, and this is being recorded on October the 16th, 2009, live on TalkShoe. For more information on the Biota podcasts, check out biota.org/podcast. And if you're interested in participating in Biota Live, there is an active chat room which is currently full of, it appears to be MSU grad students, Michigan State University, who are here because they're Avita fans, but also obviously Tierra fans and waiting for Tom Ray with much excitement. I think this is the busiest chat room we've ever had on Biota Live, and we're waiting for Tom to call in. But it's wonderful to see so many people come out and support Tom's work, and hopefully you folks will stay around for future Biota Lives and get interested in other aspects of artificial life as well, because it's wonderful to see so many people in the chat. But if you want the information on how you can participate in Biota Live, go to biota.org/podcast. It'll give you the call-in details. And for the MSU folk in the chat, if the chat goes particularly crazy and you want to actually call in and talk, the details are via biota.org/podcast. I believe also in the chat room you can see at the bottom the call-in details with regards to getting on this call this evening. Hello, Tom.
Hello.
Well, this evening's show is devoted to you and talking about 20 years of Tierra. Tom, this is your first appearance on Biota Live, and for folks listening in who, I don't know, have been living in a cave for the past 20 years and haven't heard about your work, can you describe your background and how you started developing Tierra?
Well, I'm trained as an evolutionary biologist, and I had been working in tropical rainforests for I don't know how long, I guess at the time of Tierra, more than a decade in the rainforest. And the way Tierra developed was, I was a graduate student in biology at Harvard, and I hung around the Science Center because that's where they had the PDP computer where I did some work. And they had a little cafeteria there, and one evening the Cambridge Go Club was meeting in the cafeteria, and there was a guy playing by himself, and I was curious, so I sat down and asked questions, and he was explaining the game of Go, and he was making some lifelike metaphors about the groups of pebbles growing and dying. And it turns out he was a member of the MIT Artificial Intelligence Laboratory, and he said to me, "Did you know that it's possible to write a computer program that can self-replicate?" That was a new idea to me. I'd never heard it before, so I was excited by it. And I said, "Well, how do you do it?" And he said, "It's trivial." And that was all I got out of him. But I was really excited by the idea because I imagined that if you tossed in a little mutation, you would have evolution. And I imagined a whole phylogeny of these things evolving inside the computer. Well, I didn't know much about computer. I used the PDP and typed in the keyboard and looked at the screen, but I didn't understand how you could write a self-replicating computer program, and it took me 10 years to get the insight to do it. That happened, this meeting with the fellow from the Cambridge Go Club would have been in the late 70s. Then when the personal computer came out, and I wasn't in the first wave of purchasers, but eventually I needed a computer when I was working in the field in Costa Rica, so I bought a cheap laptop computer. And I got the Borland debugger, and that was my window into the machine, because it would show the code segment and the data segment and the CPU registers. And when I saw that, then I realized that I could do it, or at least I could see the physical representation of the computer program, which I'd never seen before. I mean, the code segment, that was critical. And so I was all excited. That idea that had come to me 10 years before had just kind of, had to sit in there, frustrated basically, until I saw this Borland debugger. And then, I really wanted to do it. But by this time, I was approaching tenure at the University of Delaware on the strength of my work in tropical plant ecology. And I just knew, because the idea was so compelling, that if I was to allow myself to do this, I would fail to get tenure. It would just be a disaster. So I didn't allow myself to do it. I stayed focused on my plant ecology. But I did start communicating with people in the field. I think that was in the era of news groups, and I poked around and communicated a little bit. And somebody told me about this first Artificial Life conference that had been organized by Chris Langton. And that sounded exactly right. So I went out and I ordered the proceedings, and just loved Chris Langton's introductory chapter. He really laid it out. That was exactly what it is that I wanted to do. And I had mixed feelings about that, because, I wanted to do it, and here it was. Here was the book, and here was the conceptual framework. But by the time I got, to the end of Chris's introduction, I understood that it was, at that stage of the game, it was more of an idea and an objective than a reality. So there was still work to be done, and I communicated with him, and of course it was a new field, and so he was open to newcomers, and he arranged for me to come out to Los Alamos National Laboratory and meet with a group of people out there who were working on it, and that included Doan Farmer and Steen Rasmussen and Stephanie Forrest, and I don't think Melanie Mitchell was there at the time, but, it was a pretty core group of the original players in the field, and there was about 10 people sitting around the table in a discussion, and I was laying out the idea of working with machine code and mutating it to evolve it. Steen was really the only person who could buy that idea, because he was working with a similar system at the time. I think he called it Venus. He was more interested in the issue of the origin of life, so he was stirring randomized soups of machine code and trying to get self-replication to emerge. I wanted to start with replicators and just look at the evolution that could follow. But the rest of the group thought this was pretty hopeless, that you couldn't mutate machine code and expect to get anything but garbage out of it. And they raised the issue of brittleness, which I hadn't thought about before, but it got me to thinking about it. And so I compared the machine language to the genetic language. The genetic language could be looked at in a couple different ways. There are 4 bases that work in groups of 3, so there's 64 codons, but it's a degenerate code. It translates into 20 amino acids. So the size of the alphabet could be 4 or 64 or 20. Look at the machine code. It's a much larger alphabet, if alphabet is the right word, but the number of different instructions is larger in the sense that primarily because the actual core machine instructions tend to have the operands embedded. So if you look in the code segment, you'll see some bits dedicated to determining what the operation is, and then some bits determined to dedicated to determine provide space to specify the value that's being operated on, or it might specify a register address. But in any case, these numeric operands or addresses are embedded right into the machine code, and they alone make the size of the set astronomical. A single instruction might be 32 bits or more when you include the operands that follow the specification of the operation. So, not knowing whether it really mattered or not, I thought the first thing to do was to reduce the size of the instruction set to something in the range of the biological number of 20 to 64. And just throwing out numeric operands was an easy way to do that. But then I needed to replace them with something. And basically I had my machine instructions implicitly operate on values in the CPU register rather than values in the code segment. That's part of the solution. But then there's the issue of how do you control the jumps, because a loop in the code needs to specify where to jump to. That could be done with the values in the register too. But I came up with the idea of patterns of no-ops. I had introduced two forms of no-op, no-op 0 and no-op 1. And a jump would be followed by, say, no-op 1, no-op 0, and that would jump to the complementary pattern. 1-0's complement would be 0-1. So it would jump to the nearest instance of no-op 0, no-op 1. So I made those innovations in response to the concerns that the group at Los Alamos expressed about brittleness. And, back to the tenure thing, I didn't allow myself to sit down and code because, probably a lot of you code and you know how absorbing that can be. I didn't allow myself to do that until I submitted my dossier for tenure and promotion. And probably the same day, I sat down to code Tierra. And didn't take long. Within a couple of months, I think I'd submitted the dossier in October and had it running by January 3rd. It was running and crashing by the end of December. But I created a virtual machine with an operating system that managed a population of programs, with each one with its own virtual CPU and memory allocation. I had to have a memory allocator, and the idea of memory allocation was kind of an analogy or metaphor of the cell membrane. The idea being that without having a membrane to protect the internal domain of the individual organism, they would just ride all over each other. It would just be chaos of some sort. Though it's possible it might have worked that way. But in any case, I started out with memory allocation as a kind of I call it a semi-permeable membrane, because in terms of read, write, and execute protection, the membrane only provided write protection. So they were protected against other processes writing on their space, but they weren't protected against other processes reading or executing their code, which turned out to be important in the end. So in October of probably 90, I started writing, and I have a note here as to when that actually started running. It was January 3rd of 1990. Okay, so I must have started writing in October of 89. And what happened on January 3rd was that I guess that was when it first started running without crashing. It was just a question of getting the bugs out to where it could run. And I was at that point, I didn't have any observational tools. I actually used the debugger itself as the observational tool. And so I could run it, and I could run it in the debugger. I could step through it in the debugger, run it and stop it, and look at what was going on. And right away, I saw the emergence of these parasites because the seed organism was 80 bytes long, and these things that were 45 bytes long appeared. And by looking at what was going on in the debugger, I could tell that they were executing each other's code. So that was the beginning of the emergence of ecological evolutionary dynamic, which was really, the key thing about Tierra. That was what made it interesting and exciting. There was a period of evolution that occurred, running at the speeds I was at that time. It took a while to learn that was a transient phenomenon that after some time would lead to a stasis where, because I was giving equal time slices, which was sort of the energy metaphor, to all of the replicators in the population, there was a strong selection for optimization so that they can replicate with fewer CPU cycles. And over evolutionary time, the algorithms would get smaller and smaller. The replicators would get smaller and smaller until the original 80-byte replicator could shrink down to less than 20 bytes and still be fully replicating. And there was some limit on how small they could get. I think it was in the teens. But once they evolved to that point, there wasn't anything interesting going on anymore. They lost the ability to have ecological relationships before they completed the shrinkage of the genome, and then you would just end up with a population of sort of basically neutral mutants on whatever was the smallest thing. And you wouldn't always end up with the same endpoint, but you would end up with an endpoint. Some of the endpoints were interesting. Sometimes they had interesting optimizations, not just shrinkage of size, but they unrolled a loop, which was a clever optimization. It was nice to see it emerge through evolution. And actually, I'd never heard of that thing before, since I didn't have much experience programming at the machine code level. My training was in biology, not computer science.
In terms of the experience with regards to the Santa Fe Institute, were they running Core War? Was that something that you looked at early on or had a sense of, or is that a community that you became acquainted with after creating Tierra?
No, I looked at that before Tierra. That was, I think I had seen it in Scientific American somewhere along the line. And so when I was sort of in the period between seeing the Borland debugger and actually doing it, I was looking around, I was reading. I just didn't allow myself to code because I knew that I would get sucked into that. But, I certainly allowed myself to think and read and research and communicate. And Core Wars had some relevance. Core Wars didn't have the cell membrane, and it was pretty brutal. I guess that was part of, what led me to think cell membrane would be important. And of course, they weren't replicators in Core Wars either, but it was certainly a related phenomenon, and so I studied that.
And this was also something that I guess the folks at Santa Fe were building on. You mentioned Steen Rasmussen developing a soup. That was, I'm assuming, based in some regard on Core War as well.
I don't remember whether Steen had looked at Core Wars before developing his Venus model, but he probably did.
Anyway, I interrupted you, so please continue with the early history of Tierra.
Well, I had paused actually to induce an interruption.
Oh, very good.
That sort of represents a summary of the first phase of Tierra, and then I moved to ATR in Kyoto, Japan, where I got onto the second phase of Tierra, which was recognizing that there was a stasis there and thinking about what was needed to move forward. Well, as a biologist, I was intrigued by the Cambrian explosion because when you kind of look over the history of life on Earth, it was pretty boring before the Cambrian explosion. Just, essentially, the Cambrian explosion, in a meaningful sense, represents the transition from single-celled to multi-celled. And out of multicellularity, almost everything, macroscopically interesting in biology emerges. So anatomy, physiology, the neurobiology, even ecology and behavior, not to mention aesthetic beauty. All of these things are post-Cambrian explosion phenomena, and what you had before that would be interesting to a microbiologist, but not so interesting to most people. So I figured there were some clues in there, and as I analyzed it, what it was really about was not so much the origin of multicellularity, which could simply be many cells of the same type stuck together into a blob, but more meaningfully the origin of differentiated multicellularity, so that you had different cell types and different tissues specialized for different functions. So you could get nerve cells and heart cells and muscle cells and skin cells and so on. That was really a key aspect of the emergence of complexity that came out of the Cambrian explosion. So I wanted to basically model that in the same way that I had modeled sort of the origin of evolution. See, Steen was interested in the origin of life itself, so the origin of replication was what he was targeting. In the original Tierra, I was targeting the origin of evolution by starting with a replicator, seeding it with a replicator. Well, in the same spirit, I didn't want to get multicellularity to spontaneously emerge out of Tierra. That would be like getting replication to spontaneously emerge out of Steen's soup. What I wanted to do was seed it with the most primitively differentiated form possible, which would be 2 cell types, and see what happened from there. Well, so what is the metaphor? What is the analogy? I figured it had a metaphor with parallel processing, and so I was real interested at that during that period in parallel machines, and ATR tried to have at least one of each of them. So we had quite an interesting collection of parallel machines there to play with. And Kurt Thierling from Thinking Machines came to work with me. Thinking Machines sent him, and we got it running on the CM5. But the real metaphor and the key thing was that we seed it with something that can start with a single CPU, but we have now introduced to the instruction set a split instruction that allows a single CPU to split into 2 CPUs. They can share the same code. So we would have a single genome, but we could have multiple CPUs executing them. And it's code, so we can think of, or in fact there exist different functions in this code, subroutines that call each other. And so at some point these 2 CPUs would come to an if statement where they would jump to different locations in the code, and they would then be differentiated. They would be executing different parts of the code. To me, this was a metaphor of gene expression. So executing different parts of the code is the same as expressing different genes. So our body contains tens of thousands of genes, but a specific cell doesn't express all 10,000 genes. It expresses a subset characteristic of its cell type or tissue type. So this was the biological idea that I was developing. And I figured that the simple soup that original Tierra ran in, which was a 60,000 byte soup that held about 300 of these 80 byte creatures and their daughters, that environment probably didn't have enough complexity to favor a more complex organism. Well, this would have been in the mid 90s, and the network was sort of coming into its own, and so I got the idea of having this as a distributed system on the net, sort of SETI style, that Tierra could run as a background process on many machines, and the digital organisms would have the freedom to migrate between machines. And I imagined them foraging on the network for CPU cycles and memory space to live in, and cycles as energy. And I could envision them migrating around the planet on a daily basis, staying on the dark side of the planet where there are more free CPU cycles, imagining that in the day the CPU cycles would be occupied by their human users and the machines would be more idle at night. That was the kind of thing I envisioned. I also envisioned a fairly large region of cyberspace that could have a complex topology of its own that these digital organisms would need to navigate. So the idea was both to challenge them with a richer, more complex environment, and to seed the system with primitively differentiated digital organisms and see what happens, similar to the original Tierra experiment, just on a different level. So I designed a new ancestor that had two tissue types. One of them was basically the original algorithm, which is a replicator. The other tissue type was of a sensory nature, and what it did was to ping other machines, and we created a special Tierra ping algorithm where they're provided with a list of IP addresses of the machines participating in the experiment, and they're able to go down that list and ping machines on the list. And when the ping comes back, it brings some information about the state of that machine. So that information includes the size of the memory allocated by the Tierra process. So that's how much memory space is available on that machine. It includes an index of the speed that Tierra process is running. So how fast is it executing Tierra instructions? That's a measure of the energy level. And there was a bunch of stuff that we threw in there just to make available. I think it included the number of Tierran organisms on that machine, and I don't remember all the other stuff that was in there. So they could get this information, and the seed algorithm would get some of these. It would get maybe 8 of these and do a sort of a series of binary pairwise comparisons and end up with the most attractive machine out of the 8 based on, I think it divided the number of organisms into the speed at which Tierran instructions are being executed. So it was searching for the best allocation of cycles per creature on the machines that it's looked at. And this all was in a loop, so it would do that analysis. It would pass this information to the replicator, so when the daughter was born, it could be sent off to that machine, the best looking machine, and then it could produce another daughter while it's looking at other machines, and it would always choose the best machine out of all those that it had seen. Well, we got a number of institutions around the world interested in this idea, and they allowed us to run the process on their machines, and it turned out to completely consume their bandwidth, which got a lot of people pissed off. some institutions had trouble communicating when it ran. Others got bills for the bandwidth, which was very unfortunate. So we decided that wasn't a good idea to try to run it on a global scale, which is what we had done. And we pulled back into the institution of ATR, and we were able to get over 100 machines in-house to run it on round the clock. And so that was a compromise, but it was something that didn't get people pissed off. And the results have been interesting, but the analysis is unfinished, actually. It's not easy to analyze machine code. Anybody who's worked with a version of Tierra or derivative knows that. It's hard enough to read machine code that a human wrote, but when evolution has had a hand in it, it's all the more difficult. But now we're not just talking about regular machine code, but multi-threaded or parallel machine code written or altered by evolution. This is really difficult. And Joe Hart and I and my assistant, Chinmei Shi, have developed a number of tools that allow us to examine the structure of these organisms. Some very nice tools examine the differentiation pattern of the organism as a tree-like structure. It's complicated. Part of the problem is that I moved on to something else. I don't do this kind of work anymore, and so it remains unfinished. But I can say that that experiment produced what I was looking for to some degree, which was the origin of new tissue types and even new genes, which was something I wasn't specifically looking for, but it's part of the package. Some of the there was a little piece of code that copies a block of data from one location to another that was called by both tissues. So the replication code uses it to copy the genome from mother to daughter. The sensory code uses it, I guess, for the T-ping data to keep the best-looking T-ping data in a certain location. But these applications of that algorithm are different in magnitude. The replication of the genome involves copying a lot of data, and the replication of the T-ping involves copying a little bit of data. So one of the things that happened was that piece of code was duplicated, and then the different tissues used different copies, and then they evolved to be different, quite different. So it was an example of the origin of a new gene through duplication and divergence. And we also got, the key thing really was new tissue types. So it was groups of CPUs that executed different subsets of the genome. So I don't really know how far it went. I published some papers illustrating these results, but we have a large archive of data which is only partially analyzed, and that's kind of where it stands today.
And did Joseph Hart continue to do this work? Was there a few people that were left in Japan to continue this work?
Joe's still there. He's probably listening, and he does continue to work with Tierra. It's not his job to work with Tierra, so he's spare time working with Tierra. He, has some ideas of his own and does some experiments, adds features to Tierra, tries things. I do have two graduate students working in this area, though. Jia Xiao is working with Tierra, and she's, like many people in artificial life, interested in the big question of how does evolution generate increases in complexity. And what she's doing is using predation to introduce an evolutionary ecological dynamic into Tierra. And she's sort of at the stage now, well, she had to alter the instruction set to essentially allow the act of predation, which in her case means taking CPU cycles away from another organism, a little bit like the hyperparasites that had emerged spontaneously, but this more by design. And so she seeds Tierra with some individuals designed as predators and some individuals which are just regular Tierran replicators, which we can call prey. And she's at the stage of figuring out, with evolution turned off, how to get ecological stability in that system. And she's just completed that phase of the work. And so she's ready now, in the coming near future to be able to turn on evolution and see what happens to an evolving system like this. So, she's betting on the evolutionary ecological dynamic as a method of getting increasing complexity. My other, Gia is a PhD student. My master's student, Esther Lowe, is, yes, not really Tierra, but it's kind of my next idea along the lines of doing a thing like Tierra. My current research is in neuroscience, and I study neuromodulation, neuromodulatory receptor systems, and I'm understanding their role at the level of mind. But in learning about neuromodulation, I appreciated that in computer science, where we do, let's just say, neural nets, the phenomenon of neuromodulation hasn't been very well embraced, recognized, but it seems to be overwhelmingly important in complex brains. So I think that's an unfortunate situation for computational models of neural function, that they tend to leave this critical phenomenon of neuromodulation out. So my sort of next Tierra-like idea, well, I could characterize Tierra as transferring the phenomenon or process of evolution from the organic to the digital medium. So then the next Tierra-like idea is to transfer the phenomenon or process of neuromodulation from the organic to the digital medium. Well, this is what Esther Lowe is working on, though as a master's student, the goal isn't implementation, but conceptualization. So she's done a literature review, both of the biological literature on neuromodulation and the little bit of neuromodulation that has been introduced into computer science, review that literature, and then with a broader view than the state of the art in computer science, talk about how to introduce neuromodulation into the digital medium. I would say that the people who've done it in computer science looks like they never did a full review of the phenomenon of neuromodulation. Rather, what happened was they heard about a specific kind of neuromodulation, like NO, the nitric oxide, which is a gaseous neuromodulator, and then they would implement a model of that specific form of neuromodulation. But there doesn't appear to be anyone who's broadly thought about neuromodulation and how it can be transferred to the organic digital medium. So that's what Esther is doing. And as I say, it's at the level of conceptualization, though she's going to go into some specific, describe some specific ways of implementing it in digital systems, but though she's not going to build those systems.
Could you give a single paragraph definition for neural modulation for folks listening in?
Well, we can think of the information in the brain as flowing through ion channels, which are fast pathways. And there are excitatory receptors and inhibitory receptors, or excitatory neurotransmitters and inhibitory neurotransmitters. And one could think that would be enough to build a brain with. But these basically two forms of ion channel neurotransmission, the excitatory and the inhibitory, which is what basically all neural networks are designed around that. These two forms are surrounded by over 300 modulatory receptor types, which alter the properties of the ion channels and the behavior of neurons in very subtle ways. So, for example, if you have a neuron that has the characteristic of firing in bursts, it doesn't have to fire in bursts. That's just one way that it can fire. But if you have a neuron that's firing that way, that's its character. A neuromodulator could alter that character so that it doesn't fire in bursts. It may fire in a more steady pattern rather than in a bursting pattern. So, at the neuronal level, that's what neuromodulators are doing. My research is at the level of the mind itself. So, at that level, what neuromodulators are doing is the neuromodulators are implemented through receptors. There's a large family of receptors called the G protein coupled receptors, which includes the odor receptors. But in humans, there are about 300 different types of odor receptors and about 300 different types of neurotransmitter receptors in this family. Well, each one of these neurotransmitter receptors implements what I call a mental organ. And it's defined as the population of cells bearing that specific kind of receptor. So serotonin 2A is a specific kind of receptor. So all cells that bear that receptor would be a mental organ. And different mental organs mediate non-overlapping domains of human experience. So, for example, happiness and joy, compassion and forgiveness, logic and reason, consciousness itself, the sense of self. These are different domains of human experience which are mediated by different mental organs which are defined by different modulatory receptors. So, I've talked about it at the level of the neuron, changing the character of the behavior of the neuron, and now I'm talking about it at the level of the mind.
So, in terms of a Tierra-like model for this, is the idea to make it multidimensional, or would it still be relatively linear in terms of modeling this kind of behavior?
I don't understand the question.
So I'm interested, as you're moving into this new form of simulation, is the actual physical locations, the contacts between the neurons in a kind of three-dimensional form going to be an important component, or as you have taken Tierra, is it going to be a more abstract model that is less representative of the physical?
Well, I probably can't answer that question because I'm not myself involved in that work. As I said, Esther Lowe is doing a conceptualization of it. What I want ultimately for Esther to do is to publish a review of neuromodulation for computer scientists working with models of neural systems, to lay out the nature of modulation, the different forms of neuromodulation, and give some examples of how these different forms of neuromodulation could be implemented in conventional neural networks, for example. I'm not doing that. I'm working, I'm wet now. I'm not doing anything myself in the digital domain. It just happens that my two graduate students are both working in that area because I haven't established myself. I haven't published anything in the new area. Excuse me. I just had my first paper in that area accepted contingent on revision today. So the work is just about to begin coming out, and nobody knows I'm doing it. So of course I don't attract students in this area. My students are attracted by my previous work. So I can't really answer that question. It's the idea came to me as another Tierra-like thing that I could do, but I wasn't as excited about it as I was the original Tierra, and I don't know that I'll ever get around to actually doing it. But at this point, I'm too involved in the wet work and getting it out to contemplate actually getting involved in the digital transfer. Once that work is done and it's out, then I'll be in the position to think about, do I really want to go ahead and do the digital transfer in this domain? I don't know yet.
Yes, it's an interesting time for artificial life currently, because I'm not sure if you followed Larry Yeager's recent development with Polyworld, but he's opened up various components. So, for example, I put my Noble Ape cognitive simulation in Polyworld and taken out his sea creatures cognitive simulation and put it in Noble Ape. And there are a number of these kind of projects that are going on currently with the kind of artificial intelligence in a simulated environment version of artificial life. And it interests me that this is really a period where a number of the kind of artificial life simulators, Critterding is another one, which is a new one based on Polyworld in some regard, but with a heavy kind of neural network component at its core, seem to be exchanging neural networks and exchanging broader ideas to kind of competitively hybridize what's come through artificial life, or this particular area of artificial life. So you're talking about this at a wonderful time for the artificial life community, because these are the kind of simulations in terms of a drag and drop view of consciousness where you can put them into various pre-existing simulations. So, I mean, certainly motivate your master's student to investigate these areas as well, because super intelligent sea creatures, for example, would be wonderful to see with the kind of neural network descriptions that you're offering. But I wanted to talk a little bit about Avida and how Avida fits in with Tierra, and whether whether you see folks maturing of Avida in directions that you're talking about in terms of how you would have liked to have taken Tierra in the future, and just a kind of broader narrative about the linking of the two programs.
Well, I think it's great stuff what they're doing. Although I don't work in this area, I still get papers to review. I review quite a lot of papers in artificial life, both for conferences and journals, and so I see some of what's going on for that reason. And I know that the people working on Avida are really good people trying to solve the hard problems. And more power to them. I don't think anybody has really had the success, the ultimate goal of creating a system that spontaneously shows large increases in complexity. I think that's what a lot of people are working towards. I mean, not everybody, because some people are more interested in, say, computational intelligence than this kind of evolutionary complexity. But I guess the Avida group is has similar goals to what I was working towards and what my student Jia Xiao was working towards. So I'm only marginally up on what they're doing, but I, definitely feel that they're continuing to try to do the kinds of things that I was trying to do, and so I'm happy. In fact, as one of the reasons I was able to walk away from Tierra was because walking away from it didn't kill it. Other people are working on derivatives of it, and so it continues to live even without me being involved in it.
And in terms of, we've talked up until now about 20 years of Tierra and kind of your future work, but in terms of the artificial life community, obviously you've been a part of the artificial life community for 20 years or so. would you like to give a description of the kind of ebbs and flows that you've seen in the community as a whole, and what you think will come into the community in the future?
Well, I guess I think many of us, I recall having to explain this to Jia Xiao's thesis committee, which are zoologists who don't work in the field of artificial life for the most part, that it's kind of a rite of passage in the field to attempt to create a system that shows open-ended evolution with significant increases in complexity, seemingly able to continue as organic life has done. So it's a rite of passage to attempt to do this and to fail. And my goal was to convince her committee that it's okay for her to fail, because that's what we all do in the field. And I guess that's how I see it is that we, at least for the part of the field that has this particular goal of open-ended evolution, I'm not aware of anybody having achieved it. We do things that, by comparing with and without this thing, we can show that evolution is better with this thing than without it. And, it's a little bit like genetic algorithms and evolutionary computation field, trying all different things to improve the evolutionary process. But, outright open-ended evolution, I don't think anybody has got it or anywhere near it. Tierra looked promising for a transient period, and I guess many systems have that quality. But the big goal seems to remain elusive. And I suppose, that may be one of the reasons why I moved on. I didn't want to spend the rest of my career beating my head against the wall when I had another compelling idea that I wanted to work on.
But certainly emerging consciousness from simulated agents, I would consider part of the contemporary artificial life community, and this seems to be what you're describing as well. Do you see the artificial life community in terms of definition spreading a great deal from, the late 80s when you first entered the artificial life community? Or do you see it really, as Mark Biota does, as a kind of continuation of Chris Langton's original definition of life as it could be?
Well, I'm not going to be in quite the position that Biota is to comment on the state of the field, because apart from the reviewing that I do, I'm not watching it. I was, I guess in April, I went to an artificial life session. Christopher Nahanev invited me to speak on evolvability, and I said, well, I would just make a fool of myself if I got up there and spoke on it, because I haven't thought about it in years. I don't keep up with the literature, and he kindly allowed me to just speak on my latest research. It was very well received, and I did have a message for the community, which was sort of the good news and the bad news of what I'd learned about the mind. This was in a IEEE meeting on computational intelligence, and it was a session on artificial life within that larger meeting. So my message was for that part of artificial life that's interested in intelligence and its emergence. What I've learned studying the human mind is that it has a major division between two domains. There's what I call the cognitive domain, which is logic and reason, and then there's the affective domain, which is kind of emotions and feelings. And the cognitive domain is the newer domain, the more recently evolved domain. It's maybe uniquely human, but it's built on top of an ancient and actually more complex affective domain, which is the core of our being. And we're really nothing without it. And the bad news part of it is that I think that most of the computational work on intelligence is limited to attempting to emulate the cognitive domain. And I think that's never going to get us to where we would like to be, because it leaves out the affective domain, which is really the core of our being. The good news part of it is that what my work is showing is that the whole thing, including the affective domain, is not an amorphous mishmash. It's broken down into hundreds of components, specific components with specific underlying neural structures. So I can identify the component that's responsible for happiness and joy, and I can identify the component that's responsible for compassion, and I can identify the component that's responsible for consciousness itself, which actually was counterintuitive to me, because I'd always assumed that consciousness itself was just kind of emerged from the collective whole. But I've found an organ of consciousness, and that's kind of a new idea. But my point is that the good news is that there are specific neural structures for these specific functions, including the affective domain, which is what I think the computational approaches have left out to this point. So I don't think they're intractable as one might think if one didn't know about the structure of the mind, which is what my research is revealing. So for those interested in intelligence in the artificial domain, I would recommend learning about the affective domain, because it's really, it's the larger part of the mind, and it's the core of the mind on which the cognitive layer is built. And Antonio Damasio argues in Descartes' Error that we can't even reason without it, that it plays a critical role in all reasoning and decision-making processes. And he's probably right about that. So, I don't think artificial implementations of intelligence are going to get very far until they get real about the larger structure of the mind. It's not just cognitive. We have all these departments of cognitive science or cognitive computational whatever. Cognition is in the name of the department, and they're trying to create intelligence and mind in the machine, and they're leaving out the bulk of the mind, and I think that's a mistake.
Certainly. Well, our chat room seems to be largely filled with graduate students this evening, as they start to think about a possible career developing artificial life. I was looking through your list of funders on your website as part of your curriculum vitae, and there's an amazing list of potential funders for future artificial life research from there. For someone who would be looking for folks like Brig Kleiss and the Grateful Dead, as well as Sun Microsystems and Digital Equipment Corporation, IBM, in terms of your funders and the way that you've talked about artificial life in ways that would appeal to them, can you give some kind of overview about how to create artificial life which is immediately fundable?
Well, I was lucky because I got a lot of publicity, and in the 90s I gave over 200 invited lectures as a result, and I went to all the major computer companies, and a lot of them gave me money. That said, for people who are trying to survive in academics, it's a miracle I survived on as little funding as I got. Most of the funding that I got didn't provide overhead, which is what the institution really cares about. I had, there's one or two overhead-bearing grants in my entire career, and that's not good. I survived in part because of the publicity surrounding my work, and that's not going to cut it for most people trying to make it in academics. So I don't have a good funding record, frankly, and it's just a miracle that I made it to the level of full professor with my funding record. I would have trouble making it. Anybody with my funding record would likely to be denied tenure, actually.
And in kind of broader terms, I mean, you say that you no longer feel really as a member of the artificial life community, although you still do peer review related reading. But for recent graduates or folks listening in this evening who are thinking about taking artificial life in their own particular directions, do you have any advice to them?
Any what advice, you say?
Yes.
Well, the advice that I would offer was what I tried to express in my paper, something about Zen and the art of creating life, which is when you transfer something from the organic to the digital medium, don't be a slave to the organic form. Be true to the process. So be true to the process of evolution, or be true to the process of whatever's going on in neural systems. But if you're a slave to vividly representing the digital system in the organic system, to my mind, that's not really artificial life. That's a model. And I don't think that's what artificial life is supposed to be about. So what I tried to do in the original Tierra, and what I would do if I was to move neuromodulation over, would be to really think about the medium into which I'm inoculating this process, be it evolution, be it neuromodulation, and think about the essence of the process, the abstraction of the process, the nature of the medium, and how best to put the process into the medium in a way that's natural to the medium. Don't be too tied to the way that process manifests in the organic medium, because the organic medium and the digital medium are radically different mediums. And the same process is going to look very different in the different mediums. And if you elaborately represent the organic thing in the digital medium, then what you've got is a model. And again, I don't consider that to be artificial life, and I don't think it's very interesting, really.
Well, Tom, I'd like to thank you for your time, for appearing on Biota Live, and please do not be a stranger. We've had a number of guests that have appeared in the past with the view that they would have an interview and a chat and the kind of thing that you've done this evening, and they do occasionally reappear for general discussions. Have you been following Bruce Damer's work with the EvoGrid at all?
Yeah, he visited me recently, and we talked about it, so I'm kind of up on what he's doing.
Terrific. So maybe we could invite you back for a future EvoGrid-related discussion to reinforce what you just said with regards to artificial life and real life.
Okay.
Terrific. Well, our next show will be October 30th at 8 p.m. Pacific, where we will have Mark Badeau on. Mark will be talking about artificial life in industry and academia and the plans for the International Society of Artificial Life with regards to these two components, and maybe even something to do with a hobbyist as well. So the next show, October 30th at 8 p.m. Pacific, Mark Badeau. Thanks once again to Tom for calling in this evening, and thanks for folks for listening in.